Hello, my name is Alex Pellegrini, and this is my Microbiome Lab that I did with Simi and Tabby. So, a microbiome is the types and proportions of microorganisms living in a given location. A gut microbiome is the collection of microorganisms, bacteria, fungi, bacteriophage, and viruses present inside the gut of an animal. Research studies investigating the genetic contributors of type 2 diabetes mainly involve the use of genome-wide association studies focusing on identifying genetic components in a patient's genome. Research has indicated that the risks correlated to the type 2 diabetes may also involve the composition of intestinal microbiota. The gut microbiota of participants with type 2 diabetes displayed only a limited deviation from the control groups, although a decline in butyrate-producing bacteria that may be metabolically beneficial was observed. The observation suggests that a state of functional dysbiosis rather than any specific microbial species could have a direct association with the pathophysiology of type 2 diabetes. Increases in the presence of several categories of opportunistically pathogenic bacteria were also detected, although the abundance of these categories of opportunistic pathogens seem to be quite variable. The relationship is correlative. So, another studies have demonstrated associations between the diet and the human microbiome. Because the development of dysbiotic microbiota thought to be involved in the pathogenesis of inflammatory bowel diseases, diet is being investigated as an important etiologic factor. In IBD research, dysbiosis is associated with the progression of diseases. Lack of vitamin D receptors causes dysbiosis and changes the murine intestinal microbiome. The results are correlative. The relationship is correlative. So the question that we were asking is, will the difference in gut microbiome of two sets, will there be a difference in the gut microbiome of two sets of animals? So our hypothesis was that there will be a difference in the gut microbiome of two animals. For the first set of animals tested, it'll be in health, sex, and breed. For the second set, it'll be in age, health, and environment. The animals that we compared were samples ID 427 and 428. These are both species of dog in very good health. They're both indoor, had the same diet and exercise. No animals cohabiting. 427 was a golden retriever and 428 is a terrier. They had different age, weight, sex, and known health issues. The golden retriever has a hot spot, and the golden retriever takes supplements, takes antibiotics. Sample IDs 365 and 643 were two others that we compared. They were both German Shepherds. They had the same weight and sex, but differences in age. Overall health, supplements that they take. 365 takes coconut oil and 643 takes peanut butter treats. Takes peanut butter treats and jerky. 365 and 643 are different in that 643 is indoor and 365 is indoor and outdoor. Diet brand is also different, and 365 cohabits with a cat. So the protocol involved, we isolated bacterial DNA from feces and amplified part of the bacterial genome and sequenced that segment of DNA. So we isolate DNA from sterilized fecal samples using a Cajun kit specifically designed for isolation of DNA from stool. Then concentrate the DNA samples tenfold using the Zymo kit. We followed these two key steps intentionally, also quantifying the isolated DNA to estimate the purity to see if it is worth amplifying the specific region's 16S ribosomal RNA gene by using the polymerase chain reaction. And then the UV spectrophotometer to determine the absorption spectrophotometry to find the concentration of DNA samples to estimate if there is and how much contamination of proteins or RNA. For PCR, the cycle is 25 and has attention to double target molecules. Reaction target is denatured by heating and then allowed to cool or anneal for 45 seconds. Allowed for forward and reverse primers, and then extension slightly increased the temperature. So the gel electrophoresis was set to 250 volts, and for PCR cleanup we intentionally followed instructions and used Agincourt Ampere XP kit to clean the amplicon from our amplification PCR reactions. This removes components of amplicon PCR buffer and unused primers. We used colorimetric assay to determine the concentration of our amplified DNA samples after PCR cleanup, which allows for more precise DNA concentration. DNA sequencing done by the indexing PCR determines the nucleotide sequence of variable 16S ribosomal genes that we amplified from 1 million scat. So, the initial DNA quantification, we used the UV spectrophotometer, and as you can see, the units were taken in nanograms per microliter, and this ranges from 6.4 nanograms per microliter all the way to 85.1 nanograms per microliter. So then we took the 260-280 ratios, so the absorbance was set at 260 nanometers because DNA has a absorbance of 260 nanometers, and proteins, on the other hand, have an absorbance of 280 nanometers. A 260-280 ratio is usually between 1.5 and 2.2, which indicates pure DNA. So a ratio below 1.5 indicates protein contamination, and above 2.2 indicates protein contamination that's going to affect the rest of the results. So our 260-280 ratios, sample 645 and sample 428 were in between the 1.5 to 2.2.